Okay, so it's my pleasure to have two guests with us today to talk about probably a lot of wide-ranging things, but hopefully neurobiology, evolution, and lots of weird different types of animals. We have with us Peter Godfrey-Smith and Gáspár Jékely. Am I saying your last name right, Gáspár?
Jékely.
So no.
But
Yeah.
all right.
Yeah. Gáspár Jékely,
No worries.
who actually Peter I've known for a few years. He's a philosopher here at the University of Sydney, who I was very lucky to meet by a weird chance when I got back from my postdoc in the UK, and I went to see him give a talk in some strange corner of the university that I've never been to before or since. And he was talking about consciousness and split brains and octopuses. And yeah, I ended up putting my hand up at the end and asking him a bunch of questions. And since then, we've had the pleasure of kind of chatting and meeting at a bunch of different places and meetings. And Peter, for the first time, introduced me to Gaspar's work. I hadn't known of it before, and he pointed me towards it, and I really fell in love with some of the work and the ideas. Gaspar is a biologist who is now at Heidelberg University and does really beautiful empirical work around neuropeptides and really beautiful as well theoretical work around kind of why and how we formed these early nervous systems. And I guess from Mac and my perspective, in our groups, we work on neurobiology and neuroanatomy and trying to link it to behavior in very different systems, often in humans, sometimes in mice, sometimes using fMRI. But both of us are very interested in the links between neurobiology and behavior, but importantly, how kind of ideas from evolution can constrain it. And I think the ideas expressed in Gaspar's chemical brain hypothesis for early evolution are some of I guess really interesting constraints for thinking about behavior and neurobiology in animals all throughout the evolutionary tree. So we're thinking to start off our chat by asking Gaspar to give us a little overview of the chemical brain hypothesis, and then we can just chat from there.
Sure, great. Thanks very much for the invitation. So the chemical brain, just very simply, when we talk about nervous systems, we always think about the synapse as the main signaling hub or transmitter structure in the nervous system that transmits the activity between neurons. And so we have this very strong and core idea that nervous system is synaptically wired, and that's what makes it a nervous system. Um, and also several theories on the evolution of nervous system then started, okay, how do we make a synapse? How do we make a neuron and synapse so that we can make circuits and make behavior? Um, so this connectionist or connectomics view is very strong and prominent. And of course, then it is also very prominent, for example, in computer science or artificial neural networks. Everything is modeled based on the synaptic paradigm of the nervous system. But actually, the nervous system is not like a mechanically wired microchip where every connection is hardwired. And we know that, of course, there are many dozens of chemical molecules, mostly neuropeptides, that are equally important than synapses. And so I started to become interested in chemical signaling in the brain when we started to work on various little marine invertebrate larvae and just did simple experiments, just adding these chemicals to the water and see what happens to larvae. And we could induce amazing and very specific behaviors in these organisms, which was somehow not compatible with this idea that these chemicals are just there to little bit buffer or modulate the synaptic core of the nervous system, and they are just some kind of an add-on to the synaptic wiring. So um, and then we started also to dig in various directions, so including bioinformatically. So when did these chemicals evolve? Because there was a lot of work on the evolution of synaptic proteins, right? The synaptic machinery, when did it evolve? It turns out actually most of it goes back to before animals evolved. So it's a very ancient set of proteins that are there also in single-celled organisms like choanoflagellates that are the closest non-animal relatives of animals. So we did the same in a series of papers for neuropeptides, which was more difficult because these are shorter. So you need other types of bioinformatic approaches, and you also need to look at the receptors. But what has emerged over the years that actually these peptides are equally old, if not older, than the synaptic machinery. So also we found some peptides outside the animals and also the processing machinery. So these peptides are produced in neurons and they are chopped up by specific enzymes, and these enzymes go back before actually nervous systems evolved. So we had kind of comparative genomic evidence that neuropeptide-like molecules were there at the beginning of the nervous systems. So which came first then, right? Synapses or neuropeptides, and this is how the idea then developed. Well, let's examine this idea. Maybe it was not the synapses first, but some sort of chemical wiring without actually physical wiring, and through diffusion and receptor mediated specificity, then actually you can start to wire cells together, that specific cells talk to other specific cells in a multicellular organism. So that's kind of the core idea.
So can I ask Gaspar just a sort of question to follow up? So I think this is a really beautiful hypothesis, and I think much needed fodder for our field to kind of consider our assumptions. The synaptic kind of computation being a huge sort of almost fact that is accepted by the field rather than a question to ask about how this system works. So I really admire the work that you've done, sort of pushing back against that. So I have to admit a bit of ignorance here, and I don't know an awful lot about the chronoflagellates and a lot of these other really fascinating little creatures. In humans and in mammals, the main end organ effect, or the main effect that a cell would sense a neuropeptide with, would be a G protein coupled receptor. So one of these little serpentine receptors has second messenger cascades that can change the amount of calcium inside a cell and change excitability or change gene transcription and affect its behavior in the future. Is that thought to be the same type of messaging system that was existing back as the kind of chemical hypothesis was developing? Was that the idea that there was like a kind of system of chemical release, like neuropeptides, that were then sensed by cells, and then they could have a lot of end organ effects that were kind of slow second messenger cascades? Is that the kind of general idea?
Yes, that's the idea, and there is also evidence. So from our work, so we found these G protein coupled receptors, many of them in cnidarians, and we found the specific peptide ligands for those. We also did the same in these enigmatic placozoans, these neuron-less and muscle-less animals, very simple animals, just like a pancake with two ciliated epithelia. But they have
Wow.
actually many peptide-like molecules that also signal through these G protein coupled receptors and elicit, for example, contractions or changes in the ciliary activity. So these pathways go back also actually to the origin of nervous system. Some of these receptors are also there in the ctenophores. These are one of the most distant animal lineages from humans, and but there is no biochemical evidence yet how these act. And then, yes, these are very specific. So now in nanomolar range, a specific peptide activates one or two specific receptors, or sometimes more. And the peptides are expressed very, very specifically in a few cell types.
Mm.
And then the receptors are also expressed in other sets of, but very specific cell types. So you can imagine this wiring from certain cells to other cells through these peptide receptor links, and all by diffusion. For example, in the placozoans, there are no synapses. It's all through
Mm.
chemical diffusion.
And then the ligands themselves I gather, like in a lot of cases, the neuropeptides are actually the products of gene transcription and translation, right? They're like a gene product that gets expressed and then made a bunch of copies, and then that can be extruded from the cell and diffuse. And so you have this really lovely, it's almost like a lovely analogy of a cell that has all this information stored deep in a nucleus that needs to somehow tell the other cells in the network what's going on for them, what have they sensed, what's their the challenge have they found, what opportunity they found, and then they can sort of, this tried and tested method of I'm going to send a message out that only the cells with that receptor can actually listen to and know what to do with. It's actually just a remarkably beautiful system when you think of that complexity and how hard it would be to develop over time. But you can also just imagine how useful it would be. So I think there's a lot of plausibility to the idea. Sorry, Claire, I think you
No, no,
you wanted
Gaspar,
to
go.
ask a question.
No, go ahead, Gaspar, if you want to follow that up.
Yeah, so the main difference compared to the classical transmitters, right, which we think of, acetylcholine, serotonin, and so on, these are synthesized via enzymatic pathways, usually from amino acids, and they are modified. And then
Mm.
then these are transported into vesicles, and that's how they are released at the synapse. But the peptides, they are really encoded in these so-called precursor or pre-proteins, which have a signal peptide. They go to the secretory pathway, and then there are cleavage sites where basically specific
Mm.
enzymes cleave these, and these are conserved. And then there's a little bit of chemical modification, amidation, which makes them a bit more stable. And then they are released from these vesicles, sometimes at synapses, but often just all along the lengths of neurites or axons.
It strikes me more like a mise en place style of chefs in the kitchen, right? You prepare everything ahead of time, and then just when you need the ingredient, it's right there. You grab it, here we go, and it's off and ready to roll. It's really fun.
And maybe this is yeah, it's hard to know kind of when to jump in to sort of push you then further along in in the question of this kind of evolutionary development, because I guess the kind of next, cool question that comes up is that, if we had this neuropeptide system that was doing such a great job of kind of transmitting information and regulating sort of movement in these basic little creatures, why we then developed these more, I guess, synaptic wiring and things. And I'll let you explain it, but I guess my sort of thought from your papers is that it was almost this pressure to control sort of, I guess, larger animals and have more, more complex sort of movement effectors and then muscle and things like that, that kind of necessitated this development of the synapse, or the more, I guess, the synaptic and wiring diagram.
Yeah, maybe it's was also the size, but I'm not sure if it's only the size. Maybe more important was the speed of the transmission,
Yeah.
because there are still lots of nerve net-like organizations of neuronal structures, for example, in cnidarians, where there is release of the peptides, but there's no evidence for example, neuromuscular synapse in some systems. So, like in hydrozoan jellyfish, there is a nice peptidergic nerve net of a set of neurons. Not everything is nerve net. There is also centralization in cnidarians. But then the idea is that there is also peptide release onto the contractile cells without synapses. But of course, we know that diffusion, you can calculate exactly, how long it takes and how the concentration drops and how far you can reach with this diffusion. And of course, with wiring and precise synaptic connectivity, you can either reach further away from a source cell or really have a faster information transmission. And before there were any predators, I think the time was not so critical. And if it took you like 30 seconds to contract or 30 milliseconds, no, this didn't make a life or death decision. But when predation appeared at the origin of bilateria, then this whole thing escalated, and that's why maybe for the fast processes, the synapses took over. But of course, They didn't completely take over because every bilaterian has deeply embedded peptidergic, for example, hypothalamus or other neurohemal peptidergic organs. These are fundamentally peptidergic and regulate lots of slower processes, metabolism, growth, sexual maturation. All of these things don't have to happen at the millisecond scale.
Yeah. Yeah. I'm thinking as well about, I don't know, it's hard for me to know where to jump around with my questions because I read such a bunch of your papers in the last couple of days, and mostly with these animals and creatures that are lots of syllables that I can't even pronounce, but it's been really wonderful. And I'm just thinking about one of the papers, I guess, with you and with Peter, that you have the idea about the option space for evolution, and I guess more asking about this question of kind of why we even formed these nervous systems, which I guess, kind of leads us back to the neuropeptides and then the development of the synaptic wiring diagram. And I read about the distinction that you guys made in one of your papers, and I hadn't really known this, but I didn't, that there's sort of two camps in terms of why we evolved nervous systems, the sort of input output camp of that we evolved nervous systems to, I guess, control sort of effectors and sensors in the environment. And then this other kind of camp of the internal coordination type idea. And yeah, I guess I wondered maybe if that was something that you both wanted to talk a little bit about. That is something that, yeah, I don't think we've sort of thought too much about those distinctions, and I think the importance of considering both of them in the early evolution.
Yeah, I was going to ask something similar. Maybe I could frame it
Yeah.
for Gáspár in a particular way. If we think that there's a kind of orthodox model for nervous system evolution that has three features. One is it's a sort of input-output picture of what the early function of nervous systems was. It's responding to sensory input. A second feature is that within the input-output story, reafference is not a big thing at the early stages. The whole business of reafference is an issue for more complicated animals, and we don't have to think about that. It's just sensing from without reafference being a factor. And the third feature of the standard model is that the early machine, the early device, what you're trying to build is a network that has early synapses, targeting of influence through the morphology of the cells. If that's the standard model, or the sort of the initial picture that people have, Gáspár himself and also some of the papers written with myself and with Fred Kaiser have had three departures from that standard picture. There's firstly the idea that internal coordination might be very important with respect to the first idea, the functional side. Secondly, the idea that reafference is probably a big thing from very early stages. Don't ignore it at the early stages. And we very much, I think, Fred Kaiser is the guy who's really pushed that to us. That's an idea that he really is central to his thinking. And the third idea on what kind of object is being built in the early stages, the chemical brain is a alternative to familiar thinking about that idea. So there's three unorthodoxies put against a three-part orthodox view. And perhaps like Claire, I'm wondering what Gáspár thinks of as the links between this third unorthodoxy, the idea that the chemical brains, that's what you have very early on. What are the links between that unorthodoxy and the other ones? Is it just a totally separate set of questions? Is there some way in which they work together? How does the whole picture feel once we have the third component, the chemical brain component?
Yeah. I think this you can also think about this in terms of an option space, right? So we had this idea. This term option space from Urs Satmar, an evolutionary theoretical biologist, and we applied it to the origin of nervous systems. That let's rather than debating is it input output or this or that, let's just look at all the possibilities. And now there are more ideas than what we had originally in the paper. So we could say chemical brain and input output, right? It could be that initially there were sensory neurosecretory cells that mediated environmental cues and released peptides to coordinate some kind of effector activity that would map nicely to this option space. Or we could say that it was actually the internal coordination of the contractility that was mediated by peptides, by chemicals, and not synapses. And there are systems where we can find both of these types of signaling. So we have sensory cells, for example, in placozoans or cnidarians that are peptidergic, and we know release peptides to elicit some kind of effector mediated behaviors or feeding. And there are also systems where there are chemically mediated contractions. I mentioned the hydrozoan medusa example. So in the end, I think it's probably both of these very important early on. We know also we can trace back the evolution of sensory systems through the specific, for example, sensory receptors, opsins or trip channels. These are mechano or thermosensory channels. All of these systems go back to the origin of animals. So they must have been there. The cilia-mediated movement was there, contractility, actomyosin contractility was there, peptides were there, so everything was there. It's just maybe we don't understand the relative importance of these different systems. But I think that's now too simplistic to say that it was input-output first or coordination first or peptides first or synapses first. That may also be oversimplification.
One can imagine a narrative. I mean, this is a bit cartoon-like, perhaps. I mean, you mentioned predation. If we take that to be a Cambrian or right close to the start of the Cambrian invention, you could imagine a situation where internal coordination in the Precambrian, in the Ediacaran, when nervous systems are getting going, uh, it it doesn't have to be fast. It can be slow, as you said, and um, you're starting out with whatever resources animals have pre-nervous systems. There's a set of chemical and physiological resources that are there, and you can imagine a story in which slow chemical brain-based internal coordination functionality was first, and then scavenging arises, and then predation, and in the Cambrian that takes off, and interaction between one animal and another in real time becomes a big thing. So you have to do a lot more exterosensing. You have to get better outward directed sensors than you had before, and you have to get fast as well. So the chemical brain now becomes a little bit of a drag on your ability to keep ahead of the early arthropods, which might be running a faster machine than you if you're only using the chemical brain. So you have to start uh, mixing the electrical and the chemical signaling, and just use the chemicals at synapses, uh, because that's a way of massively increasing speed. But you don't want to electrify the whole network. This is something I talked about with Ned Block, the philosopher who's been he's been taking an excursion into speculations about early neural evolution. He's interested in the fact that you retain the chemical and the direct electrical signaling. And one idea there is, well, plasticity is furthered by, I mean, synapses with chemical signals that are controlled with gene regulation, and the cell works out which, which chemicals to make and how much to make of them. And that's a great way of keeping the system plastic, of having a way of tuning and reconfiguring what you're doing. So you don't want to electrify everything. You want you want synapses to stay around. So I mean, I have to say I'm a bit addicted to just so stories in this area, but I find myself attracted to that one. And what it's adding to other just so stories or cartoon histories that I've thought about earlier is the idea that the chemical brain is a kind of it's what you did before predation, before real-time action, as the device for achieving very early internal coordination, where that was the main thing that was the problem you had to solve very early in animal evolution. Now, again, this is very cartoony. Even the very early guys had to pay some attention to what's happening externally, but as Fred has always emphasized to us, uh, organisms without nervous systems do fine at sensing external stuff. It's not the distinctive contribution that nervous systems give us. Okay, so with some awareness of the facile nature of these stories, I offer that story.
Ah, no, look, I think that's a nice way to frame it, and I mean, this is the fun thing about phylogenetic sort of studies, right? Is that you don't have much more to go on other than plausible just so stories that then have implications that you try and test in other ways. To fast forward us, 500 million years in a blink of an eye, I think nowadays in modern systems neuroscience, we kind of often forget the sort of chemical primacy to our detriment. And one of my favorite examples in modern times is that pretty much every single mouse experiment that you see on the cover of Cell, Nature, or Science has a mouse that's been fluid restricted such that it's extremely thirsty. And that mouse wants nothing more than that little drip of water that'll come out of the little tap at the end of the little tunnel that it runs down, or as they wait over the delay and they process their little cognitive cue and they do their computation. All they're doing is waiting to get that little lick of water because they're so thirsty. So in other words, their VM hypothalamus is sitting there sending off a signal. Whatever you do, if there's water, you damn well better get it because we're really running dry. And that system basically just overrides everything. That animal will do whatever it can, solve this incredibly ludicrous set of constraints and contextual specific patterns that are, when you think of them from afar, are almost farcical what they're asking it to do compared to what it would do in its natural habitat. And they can solve these incredible problems because their chemical system has essentially said, you better well solve this problem. So I think this is just, it's just an illustrative example, but I think we kind of forget this in modern neuroscience to our detriment. And, coming from the field of human neuroimaging and now working more in the kind of cross-species systems space, it's still a really difficult problem to kind of bring the kind of real natural biology and the kind of cellular level constraint, this like really deep mechanistic understanding to the complexity of the recordings that we have. And I personally am quite motivated to try to bring more of that attention to this space. So that's more of a statement than a question. I guess what I'm really curious about, maybe from both Peter and Gáspár, whoever would like to take the question, is how do we sort of think as human neuroscientists? How do we start to think about how these systems have learned to work together, right? If the chemical system came first, and I think there's lots of very compelling evidence for that, much of which you've laid out. The synaptic system comes along and starts to play along and can do certain things that the chemical system can't do. But the chemical system is also really, really good at some things, and it's very hard-won information that the genome is unlikely to forget. These are really highly conserved genes that are sticking around across many, many, many different phyla. So I'm curious about your thoughts on how a wired nervous system learns to deal with the chemical nervous system, whether or not there'll be evolutionary trade-offs between the two. Do we think that some species are more wired and some are more chemical? I'm just really fascinated to hear kind of how you guys think about it.
Yeah. So, well, first of all, talking about human or mammalian brain, we talked about the hypothalamus as the peptidergic center. However, recent work by single cell sequencing and analyzing various, for example, cortical cell types revealed that the cortex is equally peptidergic. So Steve Smith has this very beautiful paper. He found all the
Yeah.
neuropeptides and the receptors very specifically expressed, so also chemically wired. And this actually, the way to think about this is that every single peptide receptor channel, right, forms specific connections like synapses. Cell X talks to a population of cell Y, and those are directed connections. And that's kind of one layer of a chemical network. Um, every single peptide receptor pair forms its own peptidergic chemical connectome. And then you have the synapses, you have the, let's say, also gap junctions, and then you have peptide 1, 2, 3, 4, 5, up to 100. So on average, animals have 100 different neuropeptide genes. Some precursors are also processed into different types of peptides. So you have this multi-layered connectome. And with the big connectomics electron microscopy, we are mapping the synaptic layer, but there are 100 other layers and all of them differently wired. So it's not the same cell stalk with the synapses and with the peptides. We know this where it's very well mapped. It's Caenorhabditis elegans, the nematode, where Bill Schaeffer's work mapped this peptidergic connectome together with Isabel Beetz and colleagues. So there we know that every single layer is different. So you have to think about this as a multi-layer, many layer network. And the relative importance of these is very hard, of course, to understand. But I am pretty sure if you took the cortex and took out all the neuropeptides, the mouse would be totally dead. There's no way it would function.
Just on that paper, Gáspár, that where they mapped the C. elegans networks, unless I'm misremembering, when they looked at the kind of topological overlap between the sort of synaptic and the monoaminergic and the neuropeptide, that the neuropeptide network sort of only overlapped about 5% with the other networks. So it's something really different too. It's not just kind of some neat overlay sitting atop these other monoamines and synaptic networks. It's really doing something completely different. And that really surprised me. I don't know, maybe that's something that you would have expected, but I just imagined that they would be a little bit more, I don't know, sort of nestled together or a bit more in sync than that.
Yeah. So I just briefly looked a little bit at your work and found this very nice quote from you, I think, about the metaphors, right? You had this, Claire, that some metaphors are actually helpful to think about the problem, but other metaphors are actually prevent us from thinking clearly about the problem. And I think here the metaphor of neuromodulator is that really prevents us from seeing this multi-layer structure and actually understanding how the brain works, because we just think, oh, these peptides are just there to tweak some knobs and they just little bit change this current and that current. But this work, of course, on the nematode reveals that it's completely different sets of networks. There's lots of functional work. If you take out the peptides, then most of the neuronal connections cease to exist. So there's no transfer of activity between different nodes. And so therefore, this modulatory picture is, I think, what we need to kind of rather dismiss and think of neuropeptides as core transmitters of activity in the nervous system on different pathways. So layers which, yeah, sometimes can totally not, or most often probably don't overlap at all with the synaptic layer.
Yeah.
So Gáspár, my understanding of some of Steve Smith's work, which I agree was really beautiful. It's great to see that kind of dive into the complexity of the genome and then helping us understand these kind of missing control layers that we haven't been taking into account. My understanding was that there are some neuropeptides that are, let's say, transcribed in an area like the hypothalamus and then cleaved appropriately. And then they can actually be shuttled along and treated kind of a little bit like neurotransmitters, where they can kind of be like sent to a particular area or diffusely around the brain. So a good example might be orexin or something like this that could be sort of sent around the brain to kind of, increase the excitability of a very select diffuse network. Whereas there's other types of neuropeptides that are selectively expressed in cells. So a cell, let's say, that receives a certain set of inputs will now code for a gene that makes this neuropeptide that's then locally released. But then surely then it's only really affecting its local diffusion gradient. So it's really going to be about the local microcircuit that it's around it, right? If there's an inhibitory interneuron that it could turn on to help it, let's say keep the EI balance in check if it's really excited, these little buffers for activity. And I wonder if you think about those two classes as maybe one the latter class is something that you could imagine quite easily in a smaller little nerve net type animal like a hydra or something like that. Whereas the more sort of long distance projection and kind of awakening a system feels a little bit more like like an advance that would have come with the complexity of sort of biological evolution. I'm just curious if that resonates or if you think differently about it.
Yeah, that's I don't know if that's such a categorical difference, because at least in the invertebrates where I'm more familiar with, many neurons which project far away have peptides, and these peptides fill up the entire neurite arbor of these neurons. So these long-range projections through peptidergic signals is not something that is only in for example, vertebrates. And it's probably going to be a mixture of how local and how widespread is the release. And the third case is, which is even more widespread, is the neurohemal release, right? When the peptide hormones are released into the blood, and then they can have a systemic effect. So I think this can signal at the really various scales, depending on the anatomy, but I don't think these are different specific classes of molecules that would do one or the other.
I'm I have to ask a connecting question again, but maybe in a slightly different way than the two of you have worked together, I think. Is there much known about the chemical composition of the octopus and octopus-like creatures, and whether or not there's a sort of chemical explanation for some of the beautiful features you see in like the changing of the skin? I know there's like very particular molecular mechanisms for squeezing ink packets in different sort of layers of skin, but is there a connection between those two? Oh, damn it. We just lost Peter, but yeah, I'm very
We
curious.
should,
Is there a connection?
yeah, while we are waiting for Peter, I can just say from the genomics perspective that of course all the peptides and receptors are there.
Yeah.
So the molecules are there, and what little neuroanatomy I have seen, they are of course also showing this kind of salt and peppery expression. I don't know the functional implications, but maybe Peter, if he is back, he can comment on that.
Well, we can save that one for later then. Claire, do
Yeah.
you?
I I can ask something I've been thinking about while we wait for Peter. And I don't know if it's a silly question, maybe it is, but I guess in some ways when I was reading and thinking much more about neuropeptides, it was reminding me a little bit of the sort of what seems to be this current big resurgence and interest in astrocytes. The idea that, the astrocytes are sort of doing much more than we thought. They have plasticity, and they're sort of more directly involved in behaviors than we thought. And I don't know, it seems funny to me at the moment. Astrocytes seem to be really having a moment. I don't know who's working on their PR, but they're getting a lot of attention. And in some ways, it reminds me a little bit about how you Peter just emailed and said he'll try a different way of making the internet connect. So maybe he'll come back. But I guess I was sort of thinking, of these chemical layers and the synaptic layers, and the astrocytes are probably another layer of a kind of network that's kind of interacting with these and driving behaviors. And I guess it's a roundabout way of getting to the question that I wondered about, peptidic networks. Do they show any kind of experience-dependent plasticity? In the way that, the sort of, I guess, signaling between them, or the patterns of receptors might change with experience in the way, obviously it would be different to classic synaptic plasticity, but I guess I just wondered, is there anything like experience-dependent plasticity in these networks?
Yeah, totally. There's beautiful work in Drosophila, for example, how, for example, feeding state or mating influences the preferences. For example, Ilona Kadov has beautiful work on these. There is the famous sex peptide in flies, which is basically injected um, or transferred by the male during copulation, and then this completely reprograms through the sex peptide receptor the female behavior. And many of these are long-term changes, and there is also evidence of gene expression changes, so that the receptor level changes, so that there is more or less prone to be activated by the same peptide. The nice thing about also these peptidogen systems that they can act on many different timescales. So some of them can even act on a millisecond timescale, because there are peptide-gated channels. They are known from invertebrates, from first discovered in mollusk, I think Aplysia, and then discovered in Hydra, and also now in some other invertebrates. They are not so clearly present or known from vertebrates at the moment. And then there are the G proteins with coupled receptors, which are a little bit longer timescale. And then there is the transcriptional effects, which could then change the whole gene expression program and excitability of neurons.
There was, Peter, we lost you for a little bit. An octopus
Yeah.
question had been thrown in, and then we lost our octopus guy. So
Oh, I've just changed the way I'm connecting to the internet.
uh-huh.
This is the riskier way, I
Uh,
thought, but we'll see if it's less risky in fact. What
yeah.
was the octopus thought?
There was a bit of a train of thought that Mac had. I don't know, Mac, if you want to try to.
Yeah, no, I'll try to reinvigorate it.
Yeah.
I was just really curious whether there were known chemical constraints on the many varied things that octopus-like creatures can do that are different than a lot of the rest of the animal kingdom, like the changing of the color of the camouflage, something that, chameleons can do as well. This strikes me as a very chemical kind of mechanism, and I just wondered if there was a connection there to the kind of neurohormonal sort of pathways that they've been discussing, G protein coupled receptors and release of different neuropeptides. It's a bit of an off-the-wall question, but I'm just curious if anyone knows if there are connections there.
I don't know. I've no idea. But I see the point, the interest in the question, certainly. I'm not even sure who to ask, uh, if we might ask someone about that. Um, Maybe Tamar Gutnick or someone. Um, yeah, the color changing system, it's controlled by the brain. It's got neural control over the chromatophores, and some of the other parts of the system are thought to be controlled a little differently. It's not muscle. It's not contracting or really relaxing a sac full of pigment, but something else. So it would be interesting if you had different aspects of the color changing system that ran through different routes, not just in relation to whether muscle is the effector, but with respect to what the control system is as well. That'd be very interesting.
Yeah, cool. Maybe there's a curious honors student out there somewhere that has a background in biochemistry that also wants to go scuba diving with you, Peter. So,
Yeah, yeah.
Can I do that project? That'd be awesome. One thing, okay, not octopus related, but something I was thinking about is when you mentioned Gaspar, and in the um the idea that sort of animal genomes have about 100 to 150 neuropeptide kind of, I guess, precursors or the GPCR peptide pairs. Yeah. Yeah. Why do we need so many? I mean, it almost seems, I don't know. Yeah. I don't know. Yeah. I guess, yeah. Why? It seems a lot.
Yeah. So the nice thing about the peptides, in contrast to the classical transmitters, no? To make a new classical transmitter, you need to evolve a new enzyme to embellish it with another hydroxyl or another whatever methyl group. And we know this is how neurotransmitters evolve. They become always add another group and another group. And the cnidarians, for example, often lack this final group that, let's say, in the vertebrates makes serotonin. In the cnidarians, which is the sister group, they didn't bother to embellish that molecule so much, so they just go with the precursor. And so it's hard to evolve diversity in these systems, and we only have a handful of classical transmitters. Whereas a peptide gene, you just duplicate and you just diverge, and there is almost infinite complexity. If you have a 10-mer peptide, you have 20 different amino acid possibilities in every position. So it's already infinite almost possibilities of how many sequences you can make. And it's easy also to evolve the receptors. You just duplicate the receptor and you can relatively easily discriminate. It's harder to discriminate, for example, the monoamines, and we know that receptors crosstalk. And if you do actually also in vitro pharmacology, you see that whatever octopamine activates the noradrenergic receptors at a higher concentration and vice versa. So for the peptides, we more rarely see that kind of cross activation, although it also exists. In C. elegans, we know this prominently exists some crosstalk, but otherwise they are pretty well separated what receptor goes with which peptide. And why do we need so many? I think the question also probably is biased because of our synaptic, right? Okay, we have one modulator. I understand, little bit tweak the currents, but why 100 different? Yes.
Just on the selectivity comment, I think there's actually a really fascinating molecular kind of clue here as well, which is that, as you mentioned before, Gaspar, the neuromodulatory chemicals that we typically think of are quite simple amino acids with a small modification, tryptophan or for acetylcholine, the byproducts of the Krebs cycle and a little bit of a fatty acid or something like that combined together. Whereas a lot of the neuropeptides that are commonly used for signaling in the brain and the body are these massively complicated chains of amino acids that it'd be very hard to sort of accidentally mimic. So you could imagine that if tryptophan's floating along, it might look a little bit like the binding pocket for norepinephrine or adrenaline or dopamine or something, or serotonin even. But if you're trying to match something that's this crazy long, windy protein chain, finding something that matches that and is still also sort of capable of living in the environment inside the cell outside the cell is really, really unlikely and vanishingly unlikely. So I think it's sort of another reason that you need more of these, right? Is that they're so specific and you just can't fake them. They're so weird and rare.
Yeah, larger chemical space, basically, much larger space to explore and kind of segregate for the specific signals. Yes. But why do we have so many? This is also an interesting evolutionary question. So we looked at cnidarian receptors, and it was not known whether these are homologous to the vertebrates or bilaterian receptors, flies and mice and others, or not, because you can't tell it based on the short peptide. If it's just 5 or 10 amino acids, it's very difficult to do bioinformatics, blasting, or the usual kinds of phylogenetic trees. You can't do that. So you always have to find the receptor, and we and others could show previously that the receptor and the peptide always co-evolve. So it's actually, you can follow the evolution of the receptor that describes the evolution of the peptides. So we looked in the cnidaria, we did a big screen expressing more than 150, I think, receptors, and adding all the known peptides and see what activates what. And we found many, like 20 or 30 different pairs. And then you do the phylogeny of these newly identified receptors. And there is a completely different branch from the bilaterians. So the vertebrate and like mice and fly, there's one-to-one ortholog relationship, peptide receptor for 30 different families. With the cnidarians, null, zero, nothing. And they all independently evolved the diversity in the stem of the cnidaria and the stem lineage of the bilateria. So actually it happened at least twice, and probably it seems that the placozoans also did that. So from an ancestral complement of maybe 2, 3 peptides, everyone did their own cocktail independently. So it's not just something to do with the complexity of the brain. We can say that because even the brainless placozoans did this massive expansion. It's you could imagine there could be some kind of ratchet thing, or that maybe it's just some of it is neutral, that you duplicate and kind of diverge, and then you can't go back. This may be, but I doubt that, because you have so specific expressions for all of these, which suggest really specific functions. And whenever we look at the function, then we see specific effects. So there is something about the behavioral repertoire, the cell type diversity, the neuronal diversity. By the way, neuropeptides and the homeodomain transcription factors are the best markers everywhere in every single nervous system to actually type, to tell the cell types, or for cell typing.
Like a barcode, yeah.
Barcoding. These are the best ones. You get it's combinatorial, but those are the best ones. Very highly expressed and very specific. So C. elegans and cnidarians, mouse everywhere.
And I take it, Gáspár, you'd be familiar with the prosomeric model from Neuenhaus and Puelis, that sort of thinks of, I know this from the context of mammalian evolution, but as you go through the sort of developmental stages in the embryo, you start to see the expression of particular strips of these homeobox genes that they think they're something a little bit like the stripes of a zebra, like a reaction diffusion process, where the chemical will kind of express itself, and then as it runs into its own byproducts, it starts to decrease its expression, and then you get the next line, and then there's another expression, etc. But the idea is that then that forms the gradient of the cells that form in the mesencephalon and the metencephalon and the telecephalon, that then form the basis of the circuits that allow the complex nervous system to form. So these barcode genes are both unique to different animals, but also a really helpful set of instructions for how to take the neural tube and kind of develop the scaffolding you need to build something as complex as the nervous system that we see in mammalian brains. And I think the idea is that it's conserved across other species and phyla as well. It's a really fascinating set of thinking. It kind of blows your mind a little bit to think of the complexity.
Yeah, so this is clear. These are very precisely specified, not just, so let's just put some peptides and then see what
Yeah.
happens, no?
Yeah.
Very clear transcriptional programs and very specific cocktails of peptide. And this suggests to me this idea that every neuron has its own chemical scent. Some neurons kind of have their own chemical barcode to talk to each other, and maybe if a certain type of neuron is activated, this cocktail is released, and a certain combination of receptors can sense that. So you actually, even without synaptic connectivity, you may know which type of neuron in your vicinity. This was some inhibitory neurons doing something. Let's react to that. So this chemical identity of the neuron is what emerges from all these mapping work. And this is extremely difficult to test experimentally, right? Because if it's a cocktail of, I don't know, 5 neuropeptides, who is gonna get a grant funding? Okay, I want to add 5 peptides together to my preparation and measure, right? Are you crazy? Like you just vary one parameter first. No, but it's a cocktail. No, no, no. You have to do one knockout and one. So it's also conceptually, we don't have the still experimental or conceptual tools to address this multi-layer combinatorial signaling.
Yeah, it's true. It's both to do that work, you need different methods and almost a different conceptual way of thinking, at least if you want to convince people to give you money for it, instead of this old school idea of, doing one thing, hold everything constant, one thing, that you have to think about it as this cocktail, right? Something I was thinking of that maybe brings both of you, Gaspar and Peter, back in, and I don't know if it has an easy answer because we have these nice just so stories, but I guess when we think about the chemical brain hypothesis and even these really interesting ideas of reafferents being important early on, and do you did you have an idea of the kind of experiments that we could do to test that out? I mean, is it about sort of finding evidence for these things in, in different organisms, or, have you sort of, yeah, did you have a kind of loose roadmap of where we could get more evidence for, whether what came first, this kind of, chicken or egg, or, as we said, everything kind of all together. But yeah, I guess I was just wondering your speculations on that.
Gáspár, that's for you, I think,
Yeah.
more than me.
So just one comment first. I really don't like this term just so story.
Blame Peter for that.
No, I know. You hear this or even read it in the literature. So just those story, it sounds like a bit of just hand waving. We just come up with something random. These are better called constrained speculations or transition scenarios.
Yeah.
So we are constraining with all the available knowledge as some kind of and trying to reconstruct a transition. So it's more like a historical analysis. And Detlef Arendt has this nice analogy. He said that doing like understanding evolution just from population genetics is like trying to understand the history of the Punic Wars just by reading the daily newspaper. Right. So, but the Punic Wars happened and there were specific things that happened with Hannibal coming and whatever. And so you need different methods to reconstruct deep evolution events in the past. But those things happened and we are interested. And our best way is doing the comparison, yeah, reading the daily newspaper. But then also looking at some kind of plausible transition analysis and how could this change into that and put together different lines of evidence from across the fossil record and behavior and comparative genomics and comparative biology. So the Justo story doesn't give full credit to this type of analysis.
Just to comment on that, look, I agree with that. I use the term just so story today to signal modesty in this connection, especially because in the history of our conversations, Gáspár, I think of you as very cautious about the kind of broad brush, this came before that, constrained speculation that I find almost irresistible to do all the time. You often seem very cautious about that. For example, the idea that there's a natural temporal ordering between the internal coordination function and the input-output function. That's a constrained speculation with the Cambrian as a marker of sorts there. My understanding is that, right, you've never bitten on that idea. Sorry, I'm using a lot of metaphors. You've never thought of that as as superior to other constrained speculations. You come across to me as very cautious. Are you liberalizing your attitude, or have I misunderstood your earlier attitude?
My first paper on this topic was a very input-output oriented, that the cilia motor. This was motivated by my work on the phototactic circuit of the Platineris larva, which is just a one neuron circuit, the simplest circuit that can mediate a very nice behavior. And then it's also then a plausible pathway, obviously, for a synaptic evolution from one neuron, then two neurons, then three neurons. That's obvious. And so then, okay, let's start to build a model that we start with that one neuron that controls the cilia. And then this is how my input-output oriented model was initially conceived. But then I met Peter and Fred Kaiser, and we had lots of discussions. And actually, you start to broaden your horizon. No? When Fred's papers initially came out, and I read it, oh, this is just whatever, this is gibberish, or uh, this is not a good. My model is better. No, this is the first reaction, and there are still fields in evolution where people just defend their model for like 30 years. No, what I said 30 years ago is still correct, and because of this there's no evolution, no dialogue. And that's why working with both of them was such a great experience, because actually you then really start to test your own ideas, and then you look at the evidence, and then your constraint becomes, you look at what is about the sensory history, what about the peptides, what about the nerve nets and coordination, and actually all of this goes back. So you cannot constrain that speculation by a phylogenetic signal. Therefore, that's why I'm cautious at the very base what was exactly there, because we just have very few major lineages: the ctenophores, cnidarians, placozoans, and the bilaterians, and all of them are quite different. So what was there at the base is very difficult to kind of reconstruct with phylogenetic methods. But we still don't give up, and we need more and better data. We need, for example, we don't know anything about synapses in cnidarians, their chemical nature, the transmitters, the receptors. There is no fundamental identification like in the bilaterians of a synaptic signal. We know from EM, but we don't know what's in the vesicle, what's the receptor, where's the receptor. And that prevents us from understanding the evolution of the synapse, which is, of course, very important in these scenarios.
Is it true? I might be misremembering, but there was a review that came out recently about the evolution of the synapse, and in the chronoflagellates. Is it true that they have the genetic components, or they have the genes that code for all the kind of machinery of the synapse already in them, but they don't express them, but they have all of those they code for those proteins, right?
Yeah, they express them, and they use it for vesicle secretion.
Yeah.
And almost always they have the synaptotagmins and whatever. They don't have, I think, GRIM or one or two, which are in the presynaptic complex. They don't have, but essentially all the vesicle exocytosis machinery is there.
Mm.
So but what we don't know what is in these vesicles, right? For a synapse, it's not enough to have the synaptic proteins that fuse the vesicle. That's a general eukaryotic vesicle transport machinery. You need a specific transport system to load the vesicle. Now, the evolution of these transporters is unknown, because everyone can do bioinformatics, but very few people can measure biochemically the activity of a transporter in a vesicle, right? So they would need to go back to biochemistry in many of these cases to reconstruct. You can find in genomics, like, I don't know, 20 transporters that kind of vaguely related to the bilaterian ones, but what do they actually transport? Same with enzymes. Some enzymes, not exactly the orthologs for the neurotransmitters, exist, but are they promiscuous? Can they synthesize acetylcholine or not? We don't know, and the genomics fails us at this level.
Yeah, I was trying to imagine just then how complicated the biophysical models would need to be in order to confidently recreate the emergent properties of the circuits in the embedding in the actual animal's environment, in the kind of umwelt that it lived in, and man, you'd have to basically reinvent the world again in order to figure out whether this stuff actually did what you think it would do. It's because there's so many of these sort of emergent constraints that just make this modeling so difficult.
Yeah, you can model protein structure pretty well, but really, if you model a transporter or a receptor and predict its ligand, I
Yeah.
think that for peptides there is progress, and the people can now reasonably well predict peptide receptor pairs, but it's also not as good as the pharmacology.
Yeah.
Um, and everyone is still goes to rabbits to get antibodies. To when you want
Yeah.
really specific binders, you have to go through the evolutionary experiment, and you can't predict it. So these models like AlphaFold and Boltz are fantastic, of course, but I think they lack that kind of still that predictive ability. I'm sure this will improve, but we cannot yet predict an Iderian transporter's affinity for the various chemicals, and that's what you would need to know what's in the vesicle.
I was actually just chatting with a young scientist at the University of Sydney, Chao Chua, who does a bunch of work on ion channels, and his job in the work that he does is to kind of embed the ion channel in a cell that's maybe like a blastoform or something like that, and then prove that if you put this blocker in, the cell doesn't change the membrane potential or anything like that. And I was thinking about how hard it would be then to go another level down to the actual structure of the proteins themselves and kind of guess that there was a pocket in one of these little proteins that could emit a ligand. And it's just remarkable that we've made any sense of this system at all. It's so just overwhelmingly complicated how many scales are crossed in order to have this functional organism that actually like lives in the world and adapts to the changes. And anyway, isn't evolutionary science just so beautiful as a field to think about?
Yeah, agreed. Yeah.
Did you see the paper just maybe a few weeks ago where it looks like now that the early fish had four eyes?
Whoa.
Yeah, I don't know, Gaspar, did you see it? And I guess because there's a place somewhere in China that is really great for for early fossils, and the early I don't know which fish it was, but it looks like it has had four eyes that kind of function like camera eyes, and that might have been the things that then gravitated to the middle to become the pineal eye that we have in reptiles.
Yeah.
Very cool.
Wow. How good.
Yeah, so that was always a mystery, the origin of the vertebrate eye, no? Because basically the cephalochordates Amphioxus has just one unpaired eye spot and no lateral eyes. And there was a recent theoretical paper by Tom Bad and colleagues that they talked about the pineal and the retina, that this is kind of have a common origin and very similar. So they had a model that actually is very similar, of course, but this fossil is directly in the middle of that and kind
Yeah.
of it's a nice example when you had before kind of a constraint speculation, right? A transition analysis of how you can evolve the vertebrate eye, and then bang, there's a fossil which really is completely kind of agrees with that model.
Yeah, no, I really love that. It was very, very cool. Um, Mac, I don't know if you had any more questions or things that you wanted to follow up on.
Uh, no, this has been absolutely fascinating, and I think my mind's kind of whirring with things that I want to go spend some time thinking about. The yeah, we've spent a lot of time thinking about and trying to advocate for taking seriously the neuromodulatory arousal system. So, the acetylcholine, serotonin, noradrenaline kind of side of things. In when we start to think about the complexity of the mammalian brain. We think about the corticothalamic system. How could it be modulated by these neurochemicals? And I have to admit some ignorance. I only about 6 months ago was made aware of Steve Smith's work through a collaborator, Michael Brewhouse, at the University of Washington, who'd done this really beautiful work showing that the locus coeruleus, this deep structure in the pons that is the sort of main source for noradrenaline in the mammalian brain, it projects out everywhere, douses the brain in neurochemicals, has this little inhibitory sort of structure sitting right next to it called the periceruleus that inhibits the particular subsets of that circuit. But if you go look into that periceruleus area, different parts of the periceruleus actually express very different neuropeptide receptors, and they actually selectively have different downstream behavioral effects. So if you sort of silence one of them, now all of a sudden the animal can't respond to a salient threat. If you silence another, now all of a sudden the animal doesn't do some motivated behavior like grooming or something like this. And so the idea was that you've got this incredibly good system for arousing the brain, exciting the system, making it, this part become active and this part become quiet or something like that. But behind the scenes is this really interesting little conductor that itself is controlled by this chemical system that's nuancing the different circuits to become active or not. And I just really love this idea of kind of tracking just those steps further back, because surely this neuropeptide system has got its own coordinators like the hypothalamus, like we talked about, other kinds of behavioral triggers, physiological variables, things of that nature. And I just love the idea of kind of diving into the complexity of the brainstem and thinking about all those structures and their interaction. And I really just can't wait to watch what the next generation of young scientists coming up will get to do as this data gets better, better genetic markers, better control over subcircuits, and being able to link those to the behavioral sequelae, I think is just going to be transformative for our field. And it will take us back to that kind of connection between chemical and electrical that I think is just at the basis of the nervous system. So I don't have anything really clever to ask other than to say that I really love you guys' work, and I think it's really an important kind of constraint for us to be including in theoretical models going forward.
That's a great example, Mark. And what I would be wondering about, how many of these neuropeptides signal extrasynaptically, so by volume transmission, for example, in the pons. That would be very interesting to know. Because of course, if you did the connectome of that area, those would be invisible, and then you wouldn't understand the behavior just from the synaptic connectome. So that's kind of a challenge for the people working in this brain area.
Yeah, couldn't agree more.
Before we wrap up, I don't know if you guys had anything else that you'd like to add, something that maybe we haven't covered that came to mind. Any final thoughts, Peter, or any overview you'd have?
No extra thoughts.
And Gáspár?
Well, just this closing remark that the brain is not a computer, so that's so clear. I mean, there's no chemical computers with 100 layers on top of an electric layer, so that's fundamentally misleading, I think, metaphor, right? To think of the brain as a computer, or design computers that we think should be working like the brains based on only the synaptic analogy. So that's completely misguided.
Yeah, I think we agree for sure. It's a stubborn metaphor, though. It's hard to shake.
Yeah.
Great. That was really fun.
Be a steam engine, in the whatever. The brain was a steam engine.
Yeah. Well, thank you both so much. Thank you, for being able to join together. And it was so wonderful hearing from you both, and even just hearing you both talk about the collaborations you've had. It seems like such a great collaboration. I feel like I remember I can't remember where I was, one department that I worked with where we did a lot of MRI scanning, and somebody made the joke that every MRI scanner should come with a philosopher attached to it. And the idea that, when we're running off and doing experiments and things like that, having the sort of constraints of thinking about things with philosophers, I think can be a really beautiful thing. And I think I can see that in your collaboration and the work that you've done.
Thanks, Claire. Thank you.
Thank you both so much for your time. It was wonderful to chat.
Thanks, gents.
Thank you very much. Thank you. Bye-bye.
Bye.
Goodbye. Goodbye.
Bye.